Okay, so next we're going to go to Rafael Moura, who's going to talk to us about assortative mating in space and time, patterns and biases.
It's good.
Oh, nice. Thank you very much for the opportunity. I'm really happy to be here. My name is Rafael, and I will present a lecture, Assortative Mating in Space and Time, Patterns and Biases. So, assortative mating can be described as a deviation from random mating that can emerge from a tendency in mating preference for phenotypically similar or dissimilar partners. So we can measure assortative mating using a correlation of mating pairs, of a given trait of mating pairs. When this correlation is positive, we call it as positive assortative mating. When it's negative, we call it negative assortative mating. It's very important to identify patterns of assortative mating in nature because they can provide precious information about how selection is affecting that given trait. For example, if the assortative mating is positive, maybe this trait is under exudative selection, in which the extreme phenotypes are favored by selection. So sympatry speciation is likely to occur. However, when the negative associative mating occur, stabilizing selection may favor the average type, so no speciation is expected. Associative mating can be classified also depending on the trait under selection. For example, Young et al. Classified the types of associative mating as age, behavior, chemical conditions, ecotype, genotype, phenology, size structure, and visual. However, they separate what we call as size-associative mating literature in two different types. They call size as measures of the whole body size, for example, body length, and structure as a measure of a body part, for example, the carapace width of a spider or some insect. However, here I will refer to size-associative mating as any type of measure of a body size, because they are frequently correlated, and in the literature you will rarely see some kind of structural associative mating. So researchers use two practices to investigate associative mating in nature, but they may have some problems that I will discuss here. The first one is if you want to study associative mating in two populations or two different breeding periods, you may measure the size of individuals in copula, and you can find two correlations, for example, two kinds of negative associative mating. However, if you observe the populations, you will see that the average size of individuals is different. So when you pull this data, what happens is a positive bias in the estimate of associative mating. We can call this Simpson's paradox, or in the literature of associative mating, scale of choice effect. Here in our database, we found almost 46% of the studies perform this kind of practice. A second practice is to draw broad conclusions for the whole population or even the whole species based on a single measure of associative mating. This can be very problematic, especially if associative mating vary in space and time. So in this example, you can see that there is a positive associative mating, but in another moment or in another population, you can obtain a different pattern. We found almost 38% of the studies in this database perform this kind of practice and draw broad conclusions using single measures. So our goal was was to use correlations between mating pairs to form a systematic review and a meta-analysis to evaluate the positive bias of spatially and temporally pooled data on the size-associative mating patterns, how consistent patterns of size-associative mating are across space and time, and general patterns of associative mating across and within the taxa of animals when the pooled effect size are excluded. So we follow the PRISMA protocol. I will not describe the details, but I will present here. Basically, we chose two keywords, which is size associative and size associative, and those three databases. So we obtained initially more than 50,000 results, and our last survey was formed in 20 April 2018. In total, we obtained the largest data set about size-assortative mating published to the date. So 1,827 effect size from 457 studies using 321 species. So we identified a pattern throughout the decades in which the gray bars represent new studies published in each decade, and the black dots and the lines represent the cumulative number of publications. So as you can see, assortative mating is a ongoing a field of study which is growing in each decade. When we disentangle the types of pooling practice that we classified as spatial pooling, temporal pooling, or no pooling, and even both pooling practice, we saw a tendency here that most studies publish partners without using any kind of pooling practice. However, the number of studies using temporal pooling practice has increased. So basically, we screen each study looking for Pearson's correlation coefficient, R, and when data was presented in graphs, we extracted the data points using the web plot Jitsi software and calculated our own correlation coefficient, or transform all the statistics into our correlation coefficient using the MetaWin calculator. So we use all these measures of correlation coefficient to transform in Fisher Z, which we used as effect size in our study. We use this kind of statistics because Fisher Z represents normal distribution of residuals. So we also added the weight for each effect size, which was the inverse of variance that was calculated using the number of samples. So for each, we formed several multi-level meta-analysis using the Metaphor package and included as random variables the effect size ID, the study ID, the species ID, and the phylogenetic relationships. We also added as moderator in the analysis the pooling practice, yes and no, if there is any kind of pooling practice or no. And we also disentangle the effects of different kinds of pooling practice, spatial, temporal, day interaction, and compared to no pooling practice. We measure heterogeneity using sigma squared and I squared, which measure the percentage of the total variance attributed to the random variables. So we calculated obtained the phylogenetic relationships using the Open Tree of Life data set. We extracted the metazoa tree and pruned the tree to obtain phylogenetic relationships for the species included in the meta-analysis. We also classified the animals in different subphyla. Here you can see a funnel plot with all the results separated by the types of pooling practice, and we identified a positive publication bias. But we investigated different sources of the bias, such as the dimensionality of the trait, which could be multi-tree, be unidimensional, or even dimensionless, the type of study, if the study was performed at the laboratory or at the field, and the year of publication. However, none of these moderators explained this positive bias. So we assume that this may represent a true asymmetry in assortative mating observed in nature. When we investigated the effect of pooling practice, we found a slightly positive bias in studies that perform pooling practice compared to no pooling practice, and these results remains consistent after separating the types of pooling practice, as spatial and temporal presented higher average effect size compared to no pooling practice, and there was no interaction. So there is a positive bias in pooling data. When we compare the heterogeneity of all studies included in our meta-analytic models, we found a moderate heterogeneity. However, when we excluded the studies that perform any kind of pooling practice, we found significantly less lower heterogeneity. However, the impact of this new data set on the average effect size was low. So in general, we found a moderately positive average assortative mating across all studies. We also identified, using this unbiased data set, a pattern of average effect size for each subphylum. So Mammalia was the only one that exhibited random matings, while the others, the assortative mating was positive, and Actinopterygidae exhibited the highest value. The heterogeneity of this model was the lowest of all models that we presented here. So our average patterns reported in this meta-analysis are very consistent across studies. To test spatial and temporal consistency, we performed the same type of analysis using two different kinds of data sets that presented spatial and temporal replicates. So we extracted the residuals of this meta-analysis to obtain an unbiased response variable that we included in the repeatability test. So we use as random variable the spatial temporal replicates, and to obtain the repeatability measure for each kind of replicate. So here we have the results across populations. The average pattern exhibit a low repeatability. Hexapoda and Castropoda also exhibit repeatability, but most taxa, such as Mammalia, Amphibia, and Crustacea, did not exhibit any repeatability between across populations. And between breeding period, we found similar results because the average pattern will also exceed a low repeatability, and Aves and Hexapoda presented similar results, but Amphibia, Chelicerata, and Crustacea did not exhibit perpetual patterns. In conclusion, assortative mating can vary in space and time, so assortative mating estimations should be carefully analyzed before drawing conclusions about their potential mechanisms. Focal samplings indeed provide important information about mating relationship, but they must be interpreted as punctual evaluation. So any attempt to extend these conclusions for different populations and breeding period can be misleading. Spatial temporal variation in assortative mating is more common than previously thought, and ecological conditions may have important roles in driving such variation. So if in the same species we can find a variation throughout time or space, we should look for some ecological conditions that can affect such consistency or variation. This is especially important because there is a need for new hypotheses about the underlying mechanism of spatial temporal variation in assortative mating patterns. To the date, we only have hypotheses that predict the occurrence or not of assortative mating, but no hypothesis available to explain how assortative mating should vary between populations or breeding periods. So I want to thank to my collaborators, without whom this study would not be possible, and all funding agencies. So thank you very much, and I'm available for questions. Thank you.
Great, we have a few minutes for questions.
I'll start with a question. Rafael, that's really exciting, and it sent me immediately searching off Ecology letters, where it's not quite out yet, so I'm anxiously waiting for its emergence. The question I had was that David Green recently analyzed this pooling problem using Anurin assortative mating database in American Naturalist, and found that there was actually a rather large effect of this grouping, this pooling problem. And whereas I see from your analysis, it seems that it's much smaller. So what's your take on that? Are Anurins special in this regard, or is the analysis different, or something?
Our analysis is similar, but I also cited the work of Green in this paper. But he used a data set for amphibian. And here we are using a much larger database. So as you can see in these results here, sorry, let me come back a little bit. For repeatability patterns, you can see a high variation between subphylum. So if you look for amphibia here, there is no repeatability across populations and between breeding periods. So I would expect that pooling data would be more problematic for this particular subphylum compared to, for example, hexapoda that exhibit a higher repeatability across populations in between breeding periods. So the work of Green is really nice and inspiring. It helped me a lot, but his data set is limited to amphibia.
While we wait for more questions, and I have one too, were you surprised to see, basically assortative mating be very common across all your taxa, I think you said, except for mammals? And if so, what do you think that says about the sampling bias in studies?
I don't know if I understood the question. You are trying to look for some explanation for why Mammalia has a random pattern.
Yeah, why is Mammalia so different from the other taxa, and did you find that surprising? What do you think that says about sort of the sampling bias?
I think we can look for many explanations here, but I think the difference of my data set compared to others is that I included humans here. So Young and collaborators, and also another meta-analysis published in 2019 by Janik and collaborators, excluded Homo sapiens from the analysis. So I think one of the reasons is because I included the species here, our species here. But other important questions is how mammals interpreted the significance of size in mate choice. For insects, for example, size had several implications for how they compete, they choose mate, but this is not necessarily true for mammalia. There's other conditions that can be more important than size into mammals. So I think that could be one explanation, but the focus of my study was not to explore this asymmetry between subphyla, but just to report some unbiased estimation for each taxa. Did I answer your question?
Yeah. No, I think I was surprised by that, and then I was thinking whether I should be surprised. So, but you're saying that a large part of that is because you included humans as mammals,
Yes.
or at least some part of it, yeah, which is also interesting that one taxon can have that effect. I think we have one more minute for questions.
Yes, I'm really curious to explore why, for example, some subphyla exhibit higher values of assortative mating while others do not. For example, Janik also had a paper in which he showed that the strength of assortative mating does not explain the number of species that evolved, that appear in each subphylum. Where I think his approach could have some improvements if we look for some variability in size assortative mating, which was not his goal. So I think we can discover a lot of new things here if we look to assortative mating not as a static pattern, but something that can vary.
Okay, thank you very much, Rafael.